Welcome students. I Dr. Sima Desai Fernandez from St. Xavier's College, Mapsa, Goa, Assistant Professor in Botany. Would like to take the course code that is BOC 106. Our course title is Cell Biology and Plant Biochemistry. Title of unit is Cell and its Components. Module name is Other Cell Organelles 2. Outline: the structure of Golgi apparatus, function of Golgi apparatus. Learning outcomes: describes the structure of Golgi apparatus, explains the function of Golgi apparatus. This is how the Golgi apparatus looks like. This is the structure of Golgi apparatus, where it shows the cis phase and the trans phase. Let us see the introduction. The Golgi apparatus is also known as the Golgi body or Golgi complex, or simply Golgi is a cellular organelle found in most of the cells of eukaryotic organisms. It is referred to as the manufacturing and the shipping center of the cell. It was discovered in the year 1898 by an Italian biologist, Camillo Golgi. It occurs in all cells and absent in prokaryotic cells like mycoplasmas, bacteria and blue-green algae, and eukaryotic cells of certain fungi, sperm cells of bryophytes and pteridophytes. It ranges from one to several in a cell. In plant cells, several small Golgi complex is known as dictyosomes. The Golgi apparatus is located in the cytoplasm in between the endoplasmic reticulum and the cell membrane. Let us see what is Golgi apparatus. Golgi apparatus is a stack of membrane-bound flattened sacs and are responsible for the modification of proteins received from the endoplasmic reticulum. These proteins are then transported in vesicles around the cell and hence act as the post office of the cell. This is the structure of Golgi apparatus. Golgi apparatus is composed of stacks of flattened sacs. Now, if you see the structure, these are the flattened sacs and are called as cisternae. They are arranged parallel to each other, and it contains numerous vesicles containing secretory granules with two phases. That is the cis phase. Here, the cis phase. It is called as receiving side of the Golgi apparatus, and the trans phase. This is the trans phase. It is called as shipping side of Golgi apparatus. The cis phase of the Golgi apparatus is closest to the endoplasmic reticulum. See, can you see this endoplasmic reticulum? This is rough endoplasmic reticulum, which shows the presence of ribosome. Now, this is very much close to the endoplasmic reticulum, the Golgi apparatus. So, the cis phase is close to the endoplasmic reticulum. Yes, and the trans phase is the side furthest from the nucleus, which secretes vesicles to various parts of the cell. Now here the structure shows the trans phase has secreted the vesicles. It's called as a transport vesicle to the other parts of the cell. There are a number of lumens and cisternae through which product flow. It is morphologically very similar in both plant and animal cells. It is extremely pleomorphic. In some cells, it appears compact and limited. In others, spreads out and reticular, that is net-like. Typically, it appears as a complex array of interconnecting tubules, vesicles, and cisternae. So, let us see the first flattened sac or cisternae. Now, this is the structure of the cisternae. Cisterna is the simplest unit of the Golgi apparatus and is 1 micrometer in diameter. It is central, flattened, plate-like or saucer-like. Can you see this? This is like plate-like and saucer-like. Closed compartments that are held in parallel bundles. So these cisterna or cisternae is arranged in parallel bundles, if you see the structure, or also called as stacks or they are stacking one above the other. In each stack, cisternae are separated by a space of 20 to 30 nanometer, which may contain rod-like elements or fibers. This is the magnified image of the Golgi body, which is showing the cisternae and the vesicles. Each stack of cisternae forms a dictyosomes, which contains 5 to 6 Golgi cisternae in animal cells or 20 or more cisternae in plant cells. Each cisterna is bounded by a smooth unit membrane that is 7.5 nanometer thick, having a lumen varying in width from about 500 to 1000 nanometer. The margins of each cisterna are gently curved so that the entire dictyosome of the Golgi apparatus takes on a bow-like appearance. So students, if you see this, which is showing the curved face, is like a bow-like appearance, yeah, plate-like structure or a saucer-like. So it takes like a bow-like appearance. The cisternae at the convex end of the dictyosome comprises the cis face. So if you see the convex end, this is the convex end, you see the cis phase and you see the trans phase on the concave side of the dictyosome comprises the distal maturing or the trans phase. Let us see the tubules, a complex array of associated vesicles and anastomosing tubules, which is 30 to 50 nanometer diameter, surround the dictyosome and radiate from it. In fact, the peripheral area of the dictyosome is fenestrated, that is lace-like in structure. Let us see the third part, that is vesicles. The vesicles are 60 nanometer in diameter and are of three types: transitional vesicles, secretory vesicles, and clathrin-coated vesicles. Let us see the first one, transitional vesicles. They are small, membrane-limited vesicles. It forms blebs. Blebs are like the swelling. In other words, it forms like a vesicle. Okay, so it forms blebs from transitional endoplasmic reticulum to migrate. It converge to cis phase of Golgi, where they coalesce to form new cisternae. They form the new cisternae, these vesicles. Second is the secretory vesicles. If you see the structure, here the vesicles are formed, but they are called as secretory vesicles. They are varied sized membrane limited vesicles. It is discharged from margin of cisternae of Golgi. Now here if you see, there are stacks or parallelly arranged cisternae are there. One is cisterna. Now from the margin, if you see, the structure is showing clearly from the margin, the vesicles are getting detached, and these vesicles are called as secretory vesicles. It often occur between the maturing phase of the Golgi and the plasma membrane. The third type of vesicle is the clathrin-coated vesicle. It is spherical, protuberance about 50 micrometer in diameter and with a rough surface. It is found at the periphery of the organelle, usually at the ends of single tubules. Morphologically quite distinct from the secretory vesicle. Now, this clathrin-coated vesicles plays a role in intracellular traffic of membranes and of secretory products, that is between endoplasmic reticulum and Golgi, as well as between the GERL region and the endosomal and lysosomal compartments. Let us see the function of the Golgi apparatus. Functions. The Golgi apparatus is responsible for transporting, modifying and packaging proteins and lipids into vesicles for delivery to targeted destinations. Now, as the secretory proteins move through the Golgi apparatus, a number of chemical modifications may transpire. The product enter through the cis phase and are modified by the Golgi apparatus. The modified products exit through the trans phase and are stored in vesicle. So you get that the product will first enter into the cis phase of the modified Golgi apparatus. There it is going to get modified and get exited out through the trans phase and are stored in the vesicles. They also take part in the transport of lipids and the formation of lysosomes, process materials to be removed from the cells, make and secrete mucus. Golgi apparatus is the site for the synthesis of various glycolipids, sphingomyelin, etc. In the plant cells, complex polysaccharides of the cell wall are synthesized in the Golgi apparatus. These are the references. Thank you.